Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm very excited to be here today with Dr. Suzanne Schindler, who is an associate professor of neurology at Washington University in St. Louis. I actually discovered some of Dr. Schindler's papers recently when there was a really cool one published looking at p-tau 217 in the blood and the prediction of onset of Alzheimer's. But Suzanne, you have a raft of interesting papers looking into the underlying biology of APOE and other important drivers of Alzheimer's pathology. So I'm really excited to have the conversation today. Thank you for joining me.
Well, thanks so much for inviting me. I think we'll have a great discussion today.
For sure. I want to start with a ridiculously high-level question, but probably one that you get both in your research work, but also maybe from patients you see, which is just what do we know about what causes Alzheimer's? People know about a little bit about amyloid and tau, and those aren't the full picture, and APOE is involved there somewhere. What do we know today about what the drivers are, and what are the big open questions in the field where we don't know?
So that's a great place to start. And one key point to make is that Alzheimer's is not dementia, that these are really different concepts. So cognitive impairment, dementia, is really defined as a decline in memory and thinking, that when people have impairment in activities of daily living, that's when it's called dementia. And there are many causes of dementia. So you can get dementia because you're taking a lot of sedating medications for years. You can get cognitive impairment from sleep apnea, sometimes from a variety of different medical conditions. And of course, there's many neurodegenerative diseases that cause dementia, including things like Parkinson's disease pathology can cause dementia with Lewy bodies, and certainly cerebrovascular disease. So there are many, many causes of cognitive impairment. Alzheimer's is one cause, and it often gets conflated with cognitive impairment and dementia because it's the most common in older adults, but they are different. Alzheimer's disease is defined by the presence of amyloid plaques and tau neurofibrillary tangles. If you don't have amyloid in the brain, you don't have Alzheimer's, period, regardless of what your symptoms are. So that's a really key point. And so in terms of what is, Alzheimer's, what we see in terms of the biology is that very early amyloid plaques start to deposit in the brain. And we know that people who carry this one variant in apolipoprotein E, called the epsilon E4 allele, that they are more likely to, first of all, develop amyloid plaques, and they develop these amyloid plaques earlier in life. And if you have one epsilon 4 allele, you're about 3 times more likely to develop Alzheimer's disease. And if you have 2 copies, one from your mom and one from your dad, you're about 10 times more likely to develop Alzheimer's.
Wow.
APOE is a major driver of Alzheimer's disease, and it actually, we think, does many different things. But one of the key roles is it seems to increase the likelihood that you will start to develop these amyloid plaques. So once you develop these amyloid plaques, it's really interesting. Tau, which is a cytoskeletal protein, seems to, in some way, react to the amyloid plaques. And one of the ways that it reacts is it gets phosphorylated at certain positions, including a position 217. And this results in elevated levels of soluble p-tau 217, which we can detect both in the cerebrospinal fluid and the blood. And very interestingly, we see these elevations in p-tau 217 in the cerebrospinal fluid and blood even before people have significant neurofibrillary tangle pathology. So it's a really early event that's closely linked to amyloid, not necessarily the neurofibrillary tangle pathology. And then we don't know how this happens exactly, but it seems like the amyloid causes tau to be hyperphosphorylated in some way. And the tau moves from the limbic regions to the neocortical regions. And when that happens, people develop symptoms of Alzheimer's disease. And this p-tau217 increases consistently throughout this time period when people are developing increasing levels of amyloid and tau pathology, which is over a decade or more between the initial amyloid plaque deposition and symptom onset. There's lots of other events involved in this that we don't really understand. So inflammation likely plays a key role, probably at multiple steps in the process. And, we're finding that APOE may have effects on tau as well. So there's a lot of complexity that we don't understand. And of course, many people who develop symptoms from Alzheimer's disease don't just have amyloid plaques and tau tangles. They often have vascular disease and TDP-43 and alpha-synuclein pathology. So all of this toxic slew together kind of it culminates into symptoms. So that was a long answer, but that was a big question.
Amazing and very well structured answer to a difficult question. If we stay on the amyloid and tau axis for a second, so I'll play it back to make sure I'm getting it. The amyloid plaque starts to build up, and that triggers the tau tangles that form, and the phosphorylated tau217 you're saying actually shows up in the blood, and we'll talk about your study that looked at this. Before, if you were to look in the brain, you'll see the tau tangles eventually, and that's a hallmark that you're in late stage disease, but you'll see it show up in the blood far earlier than that. How much earlier, have I got that right? And how much earlier are we talking?
Yeah, so it depends on how you do these analyses. So there's lots of ways to look at this, but the p-tau217 levels in CSF and blood change probably about 15 years before the onset of symptoms. And then the tau neurofibrillary tangles, again, depending on how you look for them, where you're looking for them, they may change maybe 6 years before the onset of symptoms. Again, there's a lot of variability in those estimates, but certainly you have had a positive plasma p-tau217 level for many, many years before we can really detect very much tau pathology with something we call the tau PET scan.
Yeah. And you talked a little bit about APOE and the APOE epsilon 4 variant. Is it right that accelerates the amyloid part of this, which then leads to the tau pathology kicking in sooner? Or how you alluded to the fact that there may be a few things going on, but is that the primary axis by which we think APOE carriers get AD? Do they get it more frequently, but do they also get it sooner?
Yeah, so it looks like what APOE does is it makes it more likely that you'll start to accumulate amyloid. But when you look at people that have reached a certain amount, kind of a tipping point of amyloid in their brain, after that tipping point, the APOE carriers and non-carriers are completely aligned. So it's not that the E4 carriers are accumulating amyloid faster than the non-carriers once they, have passed that tipping point. It's that they're more likely to reach the tipping point. And then, what is APOE doing with tau? That's a big question. And there's evidence from mouse studies, evidence from human studies that, APOE may interact with tau in some way. But I think it's still complicated. There's also interest in potential sex effects, so which may affect the relationship between amyloid and tau in women differently than in men. So there's lots of complexity here that we're still trying to figure out.
Oh, interesting. Differently how? What do we know about the direction of the effect or nature of the effect from a sex difference perspective?
Yeah, so it seems like men and women have, and of course there's a lot of emerging data on this, but relatively similar levels of amyloid pathology, but that women seem to have an increased amount of tau pathology. And why that is, what's going on, we don't know. There seems to be some kind of a disconnect between how much amyloid pathology you have and how much tau pathology you have. So it's at this point really an open question. What is, as someone named Risa Sperling puts it, how does amyloid bother tau? like
Yeah.
that relationship. And it's not consistent between individuals. Like you would think that everyone who got amyloid would a certain time later get tau. That's not what happens. So we have a lot more work to do to figure out what all these relationships are. But it does seem that things like age and sex and APOE may play a role in that relationship.
Yeah, interesting. And do you see people on ends of that spectrum where they have an extreme amyloid pathology and no tau, or an extreme tau pathology and no amyloid? Is there something we can learn from those probably rare examples where they don't coexist? And what does that look like clinically?
Yeah, so it doesn't happen very often, but it does happen. There's a kind of a famous patient who frequently talks to media, so I don't feel bad about saying his name, but it's Doug Whitney, who has an autosomal dominant mutation that would be expected to result in symptomatic Alzheimer's disease. And he's still cognitively unimpaired despite having a very high level of amyloid pathology. Now, when we look at his brain with tau PET scans, he has less tau pathology than you might expect given the level of amyloid pathology. He does have some tau pathology. So I think he's at risk for developing symptoms, but it does highlight that there's sometimes a disconnect between the amyloid and tau pathology, which is very interesting. And then there's, of course, been other cases noted of mutations in APOE, the APOE Christchurch mutation, where people can have very high levels of amyloid, but that seems not to result in what we'd expect for tau pathology and symptoms. So again, these relationships between amyloid and tau, so they're consistent in most people.
Yeah.
But there's outliers, and the outliers are actually very informative. They're telling us important information. They're telling us that APOE is involved with this relationship, which, we knew that APOE was involved in kind of affecting when amyloid, if amyloid started to accumulate and when. But the fact that it's has a role in this relationship between amyloid and tau is really interesting. And it also, gives us ideas of things we might want to target for therapeutics, right? If APOE is playing a key role in this, maybe we can target APOE and that will reduce tau pathology, which is really what seems to drive the symptoms in Alzheimer's disease.
Yeah, really interesting. It's maybe a good segue to your paper that I was talking about. We will include a link in the show notes for anyone who wants to read in detail. But you measured p-tau 217 in the blood, and you had also. I'll make sure we link your LinkedIn or your group page so that people can see some of the really beautiful animations that you've done to help people visualize this risk shift. If people haven't read the paper, could you summarize a little bit what the key findings were from that work?
Sure. And, I just note that people have kind of applied the same general mathematical framework to some different biomarkers over time, including my group. But people have been looking at this for a while. The key point is that if you just look at these biomarkers as a function of age, it's a mess. That if you look at trajectories, they really don't align. And that's because people start to accumulate these pathologies at different ages. However, the key point is once you reach a certain level with each of these biomarkers, whether it's amyloid PET, tau PET, or plasma p-tau 217, that the trajectories line up. That there's a consistent rate of change in all of these biomarkers across individuals, which is kind of surprising, right? Well, you might expect that different people would have very different trajectories, but at least for these biomarkers, which represent amyloid and tau pathology, we find very consistent changes across individuals. And why is that important? Well, it gives us a way to make it a biological anchor. So, as I like to use the analogy of tree rings, trees accumulate one ring a year. And so if it's 2026 and there's 16 tree rings, you know that the tree started to grow around, 2010, right? Because there's this consistency and what happens with the tree rings, you know when this pathology started. So that gives you an anchor point, a time zero. And that's really been a problem in many of our studies of Alzheimer's, that everybody's at such a different point in the disease that it's hard to see the underlying pattern. And so we applied these models, we call them clock models, to figure out how long does it take, what the tree rings are basically, how long does it take to get from one level of p-tau 217 to another level of p-tau 217. And then using those clocks, we can figure out, a number of points, but a key one being essentially when did they become positive or abnormal for that biomarker. And then you can anchor other events to that. So for this paper, we anchored the age at percent p-tau 217 positivity really to two outcomes. One was time to Alzheimer's disease symptoms. The other was age at Alzheimer's disease symptoms. And we found a strong association between those. So basically, you could estimate the age that someone would become symptomatic, or you could estimate the time until they became symptomatic based on a single plasma p-tau 217 measurement. The caveat, of course, is there's still a lot of error in these estimates. So they're at a level that they could be useful for research studies or clinical trials, but not at a level where they're clinically useful. Another kind of caveat to this is that there's lots of other pathologies contributing to symptoms. So we think we could make these clocks better in terms of estimating when someone might develop symptoms if we can integrate biomarkers of other pathologies. So if we can integrate biomarkers of vascular disease, for example, or TDP-43, we could somehow put these all together, we could really refine our estimates further. But the bottom line is it shows that it at least appears feasible to estimate when someone might develop symptoms of Alzheimer's disease. There's a conditional part of this. We developed these estimates in people who did develop symptomatic Alzheimer's disease. However, when we applied this estimated age at percent p-tau 217 positivity to the entire cohort, which included both people who progressed and did not progress, it was a good predictor of basically ordering who would progress. So there's a lot more work that needs to be done, but I think it can shift the conversation more from, is someone at risk or not, to when would they be likely to develop symptoms, which I think is an important shift.
Yeah, definitely. You mentioned this being potentially useful in clinical trials. How is it already being planned to be used, or how do you envision it being used there?
Yeah. So actually, I'll start with research. So we've already used similar kind of estimates for when people become amyloid PET positive and tau PET positive to align all the other biomarkers for Alzheimer's disease. So plasma biomarkers, imaging biomarkers, cognitive biomarkers, to see when they change. And it allows you to essentially make a map of overall biomarker changes in Alzheimer's. And I think that's really useful to know what are the events that occur in the pathophysiological cascade. From a clinical trial perspective, one issue is that clinical trials are usually pretty short. And if you're including a lot of people who will never progress during the study window, you have to enroll a lot of people. So utility of this is you could pick out people more likely to progress. Something that we found in our study that was a little bit surprising was that, well, maybe not as so surprising, we found that at the same level of p-tau 217, that older people were more likely to have symptoms than younger people. And that's actually not necessarily been well appreciated. And so people have kind of expected that if you were p-tau 217 positive, it would mean the same thing in someone that was older or younger. But really what we found is that if you're older and you're 217 positive, you're likely to develop symptoms in a short time, maybe 5 or 10 years, versus if you're 60 when you become positive, it may be much longer, so 10 or 20 years.
Yeah.
So I think that that's helpful for clinical trialists when they're thinking about the time frame for these studies. One thing, I think there's two take homes. One is that if you enroll a lot of younger people, you're going to need a lot larger sample size, or you could enroll younger people who have an even higher level of p-tau 217. So I think it does provide some lessons.
And with that age effect that you saw where, say, a 70-year-old with the same p-tau 217 levels, a 60-year-old, the 70-year-old is going to have a shorter window on average. Do you think that the age is a proxy for all of these other factors that you mentioned, that you've got all these other biomarkers that you're just not measuring and they're being captured in some indirect way through age? Is that, or is there something else going on?
I think that's exactly what's happening. And of course, this is a testable hypothesis.
Yeah.
So I talked about making the clock better if we can add in measures for these other co-pathologies, which really increase with age. I think we can better predict symptom onset. And of course, age reflects many different processes, some that we have biomarkers for and some that we don't have biomarkers for. But it's showing us that there's a lot of changes in the brain that affect essentially resilience to cognitive impairment. And so we have some work to do to figure out what those are and make biomarkers for them that hopefully will increase the amount of variance that's explained by these models.
Yeah. What thoughts do you have after this work and all the other work you've done looking into the mechanisms underpinning this disease in general about the most potentially effective therapeutic strategies? Because there are a lot of them, and we've had some data recently in a field where we haven't had clinical trial data that was of much substance in a really long time. But yeah, I'd love your, maybe again, an incredibly broad question, but I'd love your thoughts on the field.
Yeah, so I know that there's different opinions on this, but I really do think amyloid targeting treatments can help if you give them early enough. And I think the data certainly support that they do slow cognitive decline. Even when people have mild symptoms, I think once people get to moderate symptoms, they don't help. I'm actually really excited to see what the amyloid targeting treatments do even earlier in the course of the disease before people develop symptoms. And there's, of course, trials underway to look at that question. And then also tau targeting treatments, I think, are potentially very helpful because, of course, tau is much more insoluble tau pathology, neurofibrillary tangles, much more strongly correlated with cognitive symptoms. And a number of companies are working on tau targeting treatments. It's a little bit more challenging because tau is intracellular, but there's a variety of ways to get at it. I think that there's a lot of potential for combined treatments. So, for example, giving people treatments for both amyloid and tau. And then, of course, there's lots of other potential treatments. So we talked about APOE. There's lots of questions about what exactly you would do to APOE, though, to make it therapeutic. So there's certainly debate about that. And then there's a lot of information or a lot of interest in inflammation. The thing about inflammation is it's so complicated and it likely plays a role at multiple points in the disease, and there's many different molecules involved. So people have been very interested in the role of TREM2, but what do you do with TREM2? Do you make it go up? Do you make it go down? And it may do different things at different points in the disease process. Something about inflammation is not monotonic. So it's not like amyloid or tau where usually up is bad. Inflammation has a very complicated course. So, and then people are interested in a whole variety of other factors, everything from growth factors, trophic factors, to factors that modulate neurotransmitters and receptors. And I think it's all good. The more different ways that we're looking at this problem, the better. But what we have found so far is that the strategies that affect the defining features of Alzheimer's, amyloid, and tau seem to be the most promising. Of course, we need treatments for all these other neurodegenerative disorders as well, and people are working on that. So how do we treat alpha-synuclein pathology, TDP-43 pathology? There's lots of things that we need to target.
Yeah, I think you looked in the paper that we've been talking about, the p-tau 217 blood test. You did look at whether genetics, or at least APOE E4, adds any signal, and I don't think you found anything in that paper, right? Can you talk a little bit about whether you expect that most of the genetic signal is going to be basically captured through that test, so it's redundant in a way, or is there a sample size or power question? How do you think about that?
Yeah, so that's an excellent question. And something to keep in mind about p-tau217 is it reflects both amyloid and tau pathology. So not just one or the other.
Right.
And previously, when we looked at amyloid and tau trajectories, they were very consistent, even when we looked at E4 carriers and non-carriers, once you reach that tipping point. Before the tipping point, there is a lot of variance, which is very interesting. So it seems like APOE is having a lot of the effects really early in the course of disease and deciding, who's, for example, going to become amyloid positive. So I think that that's why we didn't see an effect of APOE, because it reflects both amyloid and tau pathology. As I mentioned, it looks like there are differences in tau pathology more related to APOE and sex that we did not see. But of course, a lot of p-tau217 is related to amyloid. So I think that mixing of amyloid and tau pathology is good in some ways because we're reflecting both pathologies, but it may not allow us to see some of the variation in tau pathology in particular that could be related to some of these other covariates like age and sex. Now, there definitely are always sample size issues, and it's possible that with larger sample sizes or different types of models, we might see effects. So I think it remains an open question.
Yeah, super. I want to get your perspective on clinical phenotyping to where, if we were to ask maybe a similar question of where is the state of the art in terms of understanding, we can use a blood test like this to understand where somebody is biochemically, but clinically, it is also a very heterogeneous disease. What are you finding is most helpful in terms of understanding not just the trajectory towards a clinical diagnosis, but some of those early signs and symptoms that somebody is progressing? I'm thinking about how we combine the biomarker data with some of the clinical data over time to get a clearer picture of where any individual patient is in their journey.
Yes, and I'm a clinician, so certainly I think a lot about those sorts of questions. So one thing about our study in Nature Medicine is we looked at symptomatic AD. So we looked at people who, if I saw them in my clinic, I would diagnose them with Alzheimer's disease. So they had an Alzheimer's disease clinical syndrome. They were biomarker positive. They didn't have a lot of features that would suggest that they had major other neurodegenerative diseases. And that's actually different from some other studies that have used this modeling technique, and that many of these other studies just said cognitive impairment that was caused by any issue. So all cause dementia, all cause cognitive impairment.
Right.
And I think those studies didn't see a kind of as clean of results because they were looking at a lot of mixed pathology. And I think because we were looking specifically at people with symptomatic AD, we really saw much more of a signal. That said, over time, we need to get to the point where our models can handle like all of these different diagnoses. That will require that we get timelines or clocks, not just for amyloid or tau or p-tau217, but all these other pathologies to really be able to understand them. And there's other questions that we didn't explore in our paper, but one thing I found very interesting is that patients who have early onset Alzheimer's disease, in our paper, we show that they need more pathology before they're symptomatic. But boy, when we see them in the clinic, once they're symptomatic, they decline very rapidly.
Mm.
So they seem to have a faster rate of progression versus older patients seem to develop symptoms with lower levels of pathology, but then when they progress, they don't progress quite as rapidly. So there's kind of this disconnect between how long it takes to develop symptoms and then the rate of progression once you develop symptoms. So there's just kind of differences in these different points and relationships that are very interesting. So again, we have a lot more work to do to explain all of these features. I just, again, seeing patients regularly, they're very interested in their prognosis.
Yeah.
They want to know what is my life going to be like in 2 years, 5 years, 10 years? And right now we don't really have clear answers. I don't know that we'll ever necessarily have clear answers, but I think data can help us to provide a little bit more information.
The point you're making just there about seeing two different things on different ends of the distribution. I'm wondering, do you think that there is some kind of multimodal or bimodal distribution where you have earlier, faster progressors and then later, and that's some underlying difference in biology, or is there just something about that it is more or less a spectrum, and the way we cut the data, we will see things on different tails when we look at them? How do you think about what that tells us?
Yeah, I suspect it's some combination of both.
Yeah.
So there's lots of interest in looking at early onset Alzheimer's disease where people develop symptoms before they're age 65. And it does look a bit different. So
Yeah.
people tend to have more atypical features. So you're more likely to see, for example, something called posterior cortical atrophy, where people essentially become blind, have a lot of visual spatial issues, and it's related to Alzheimer's disease pathology. So it's a variant of Alzheimer's, and it's much more common in in younger individuals. Why that happens, we don't know. So there do seem to be differences related to age by the clinical manifestations of Alzheimer's disease. That said, we see some of those manifestations in older people as well. So maybe a combination of that bimodal, but then there's also a continuous aspect of it as well.
Yeah. If we take the p-tau 217 as a single biomarker and we think about a world where you expand that out into the whole proteome, do you see us getting a ton of extra signal from that? Or is it likely that there's maybe 2 or 3 of these that capture a lot of the biology? What is some of the work you've done in plasma proteome told you about specifically what shows up in the blood? Because I suspect if we had the brain, we can learn a lot, but we can't do that at a population scale, at least not today.
Yeah, so work on this is ongoing, but what I would generally say is a couple of things. One is p-tau 217 is really an outstanding biomarker of Alzheimer's disease pathology, the amyloid plaques and tau tangles. So, and it's actually, when you add lots of other things into the model, it's actually harder to get an improvement in terms of connecting to amyloid and tau pathology, more with amyloid pathology. There are actually better biomarkers of tau pathology, like one called EMTBR tau 243. But in terms of symptoms, that's another matter. There's still a lot of unexplained variance between p-tau 217 and symptoms. And there's other work suggesting that you can add value to p-tau 217 in predicting cognitive symptoms. Some of that work is ongoing right now. So I do think we will be able to improve it. Obviously, now we have incredible proteomics panels that can measure thousands of proteins with a drop of blood. So I do think that we'll be able to improve on p-tau 217 in terms of connecting it to symptoms. The question is, how good can we make it? I do think cognitive tests and imaging measures will help as well. Of course, the more modalities you add into this, the harder it is to apply in research studies and in clinical practice. But I do think all of these things will add value to just a blood test.
Can you talk a little bit about the EMTBR tau 243? You published an extremely recent paper on that. That's probably the even more hot off the presses one. Can you talk a little bit about that biomarker and what it adds above and beyond the 217?
Sure. So Kanta Hourier and Dr. Randall Bateman's lab developed this biomarker called EMTBR tau 243. So it is a piece of tau that is very strongly associated with these tau neurofibrillary tangles, and it's more strongly associated with cognitive impairment, and the degree of cognitive impairment even than more strongly associated than p-tau 217. So it's a really good biomarker of both tau pathology and of symptoms, which makes it very exciting. And in a very recent paper published in Lancet Neurology, Nicholas Matson combined this with p-tau 217 and found that you could more specifically pick out the people who were likely to be clinically diagnosed with Alzheimer's disease. One problem with p-tau217 is in some ways it's too sensitive. That there's lots of people who are p-tau217 positive who don't have symptoms yet. Now they're at risk for symptoms, but they don't have symptoms yet. With EMTBR tau 243, it's much more specific. So if you have EMTBR tau 243 elevation, you are likely to have symptoms from Alzheimer's disease. That said, it's not perfectly sensitive. So it will miss some people who have Alzheimer's disease symptoms from AD pathology. So you have to be a little bit careful with it because you don't want to, miss people who are symptomatic from Alzheimer's disease. But I do think that it may be helpful, particularly in cases where you have borderline p-tau217 levels, where the patient has a lot of other clinical issues and you're wondering, are their symptoms really related to AD pathology or something else? So I think it will be helpful in select patients for sure.
Yeah, this is interesting. And I'm going to do live statistics, so hopefully I don't get this wrong, but we'll include the paper in the link. But I think the part you're referring to is within percent p-tau217 positive individuals, it has positive predictive value of 76%, negative predictive value of 90%. So basically, you're going to identify 3 out of the 4 roughly of the true cases, and you may miss 1 out of 10 of the false cases. So you could see, like you said earlier, these things may take some time to be clinically useful, but you could imagine in a clinical trial or other contexts that being an acceptable trade-off to make if you're trying to be really thoughtful about how you screen.
Yes. And how you use these really depends on the application. So for example, for clinical trials, you may really want to make certain that patients or research participants in your trial have both amyloid and tau pathology. So you could use EMTBR tau 243 for that. On the other hand, maybe you want people who are amyloid positive and tau negative for your tau treatment. Maybe you think that's the best time to intervene. It might be. And
so then are you saying you'd use the EMTBR to rule people out?
Well, you would want people who are p-tau217 positive and EMTBR tau 243 negative.
Yeah, interesting.
Yeah, so it all depends on, how you're trying to structure this.
Yeah, very cool. As we wrap up here, what if you think about the next couple of years and where the focus of your research group is, what are the couple things you're most excited about?
Well, I actually am quite excited about having new biomarkers for non-Alzheimer's disease pathologies. I think that that's something that we really need. And I do think that with these large proteomic panels, they'll help us to identify good candidates. One thing is these proteomics panels help us to identify candidates, but then we often have to make it better, tweak it a bit. So obviously we identified p-tau 181 as a good candidate, but it turns out this 217 is so much better. So I'll be curious to see whether some of these early hits we find that modified versions of them are even better. We also need good reference standards for some of these co-pathologies, like PET scans for alpha-synuclein or TDP-43 that help us to make blood tests for those pathologies. So the reference standards are really helpful. And I'm also excited about using these tools in the clinic. So I do use blood tests right now in my clinical practice, and I think it's really going to transform the diagnosis and treatment of Alzheimer's, which is really exciting.
It is. It's amazing. Well, thank you for sharing all this amazing knowledge with us and all the great work you do. I really appreciate you joining me today.
Well, thanks so much for your great questions and interest.
It was a ton of fun. Thanks, everybody, as always, for listening, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram @The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show, Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.